Bismillahirrahmanirrahim. Today we'll discuss bone. I am Dr. Muhammad Shahid Akhtar. The objectives of today's lecture is we will study bone cell, that is osteocytes, osteoblasts, osteoclasts, the bone matrix, periosteum and endosteum, type of bone, primary immature woven bone and mature secondary lamellar bone. Bone. It is the main constituent of the adult skeleton. It supports fleshy structures, protects vital organs such as those in the cranial, that is the brain, and thoracic cavities, heart, lungs, and major blood vessels, and harbors the bone marrow where blood cells are formed. Bones also serves as reservoirs of calcium, phosphate, and other ions that can be released or stored in a controlled fashion to maintain constant concentration of these important ions in body fluids. It forms a system of levers that multiply the forces generated during skeletal muscle contraction and transform them into bodily movements. Bone is a specialized connective tissue. It is composed of calcified intracellular material, that is bone matrix, and three cell types. Number one cell is the osteocytes, which are found in cavities, lacuna, between layers, lamella of bone matrix. Number two cells are osteoblasts, which are synthesize the organic components of the matrix. And number three is the osteoclasts, which are multinucleated giant cells involved in the resorption and remodeling of bone tissue. So, three types of cells. Number one is the osteocytes, number two is the osteoblasts, and number three is the osteoclasts. Because metabolites are unable to diffuse through the calcified matrix of bone, the exchanges between osteocytes and blood capillaries depend on communication through the canaliculi, which are very thin cylindrical spaces that perforate the matrix. In this slide, we can see these are the canals through which the blood vessels passes. This is the endosteum and this is the periosteum. From the periosteum, blood goes to these canals to the endosteum. All bones are lined on both internal and external surfaces by layers of connective tissue containing osteogenic cells. Endosteum is the lining on the internal surface and periosteum is the lining on the external surface. Bone cells. Osteoblasts. They are responsible for the synthesis of the organic components of bone matrix, consisting of type 1 collagen fibers, proteoglycans, and several glycoproteins, including osteonectins. So, the main responsibility of these cells, they are responsible for synthesis of organic components of bone matrix. Deposition of the inorganic components of bones also depends on variable osteoblasts. These are the osteoblasts. You see, this is the osteoblast. This is the osteoblast. Osteoblasts are located exclusively at the surface of the bone matrix, usually side by side in a layer somewhat resembling a simple epithelium. These are the osteoblasts, this one. When they are actively engaged in matrix synthesis, osteoblasts have a cuboidal to columnar shape. Just this is the cuboidal shape and this one is the columnar shape. And basophilic cytoplasm, when their synthesizing activity declines, they flatten and cytoplasmic basophilia is reduced. Just here we can see this one is the osteoblast activity is stimulated by parathyroid hormone PTH. This diagram shows this one is the osteoblast, this is the osteoclast, these are the osteocytes, and these are the mesenchyme. Having mesenchymal cells, whole is the bone matrix in which lies the osteocytes. In the cavity, this surrounding cavity around this osteocyte, this is the lacunae, and this is the newly formed matrix, that is osteoid. This lighter color area, this is the osteoid, newly formed matrix. As I've discussed in the previous pictures, this is the osteoid, lighter color, and osteocytes, these are less rounded and located within the lacunae. During matrix synthesis, osteoblasts have the ultrastructure of cells actively synthesizing proteins for secretion. Osteoblasts are polarized cells. Matrix components are secreted at the cell surface in contact with older bone matrix, producing a layer of new but not yet calcified material called osteoid between the osteoblast layer and the bone formed layer. This process of bone oppositional growth is completed by subsequent deposition of calcium salts into the newly formed matrix. Just in this diagram, you see this is the osteoblast layer, and this is the new layer, osteoid layer, and this one is the older one, mineralized bone. These osteoblast cells, they secrete vesicles, and these release into the matrix and collagen fibers. Then these areas, they got early mineralization around vesicles occur, and then matrix becoming confluenced by the vesicles. So this is the secretory vesicles in the osteoblast. The secretory vesicles in the osteoblast released in the osteoid layer where vesicles are formed, and early mineralization around these vesicles occurs, and matrix becomes confluent between vesicles, and so then the bone becomes, this area becomes mineralized and bone is formed. This area, this layer is the osteoid layer, newly forming bone layer. Calcification of the matrix is not completely understood. Prominent among the non-collagen proteins secreted by the osteoblast is the small vitamin K dependent polypeptide osteocalcin, which together with various glycoprotein binds calcium ions and raises their concentration locally. Osteoblasts also release membrane-enclosed vesicles rich in alkaline phosphatase and other enzymes whose activity raises the local concentration of phosphate ions. With high concentration of both ions, these matrix vesicles serve as foci for the formation of hydroxyapatite crystals, the first visible step in calcification. These crystals grow rapidly by acceleration of more mineral and eventually form the confluent mass of calcified material embedding the collagen fibers and proteoglycans. The next cells, osteocytes. Individual osteoblasts are gradually surrounded by their own secretion and become osteocytes. This one is the osteocytes, enclosed within a space called lacunae. This is the space in which the cells lie. This hole is the space in which the cells lie. This is called lacunae. In the transition from osteoblasts to the osteocytes, the cell extends many long cytoplasmic processes which also become surrounded by calcifying material. These are the cytoplasmic processes which extended and they are literally calcified. So this is the lacunae, and this is the cell which lies in this lacunae. An osteocytes and process occupy each lacunae and the canaliculi radiating from it. This is called canaliculi. These canaliculi, they are radiating from it. They are flat, almond-shaped cells exhibit a significantly reduced rough endoplasmic reticulum and Golgi apparatus and more condensed nuclear chromatin. These cells are involved in maintaining the bony matrix, and their death is followed by resorption of this matrix. So this is present in the bony matrix, and after the death they are absorbed by the matrix. Just a photomicrograph of brain, not decalcified and sanctioned, but dried and ground very thin for demonstration and of lacunae. These are the lacunae, and these are the canaliculi. These radiations from these lacunae, these are the canaliculi. The lacunae and canaliculi appear dark and show the communication between these structures through which nutrients derived from blood vessels diffuse and are passed from cell to cell in living bones. So these lacunae and canaliculi receive the nutrients and passes to the cells. The number 3 cells, that is osteoclasts. They are very large cells, motile cells with multiple nuclei. This one is the osteoclast. The large size and multinucleated condition of osteoclasts is due to their origin from fusion of bone marrow derived cells. In areas of bone undergoing resorption, osteoclasts lie within the enzymatically etched depressions or crypts in the matrix known as resorption base, formerly called house-shaped lacunae. So these are the multinucleated cells, large motile cells having multiple nuclei, and they are present in the edge depressions or crypts. They are called resorption base. Electron microscope diagram showing an active osteoclast cultured on a flat substrate of bone. A trench is formed, just this shape like a trench. This is a trench depression on bone surface as the osteoclast crawls along. This one is the osteoclast we show, which is crawling along the surface of the bone and on the bone matrix. Now coming towards the bone matrix. In organic matrix, it represents about 15% of the dry weight of the bone matrix. It contains calcium, phosphorus, bicarbonate, citrate, magnesium, potassium, and sodium. Organic matrix. It contains collagen type 1 and ground substance. Ground substance contains special glycoprotein which are responsible for promoting calcification. Alkaline phosphatase produced by osteoblasts also help calcification. Calcium, phosphorus, they form hydroxyapatite crystals. Significant quantities of amorphous non-crystalline calcium phosphate are also present. The surface ions of hydroxyapatite are hydrated and a layer of water and ions form around this crystal. This layer, the hydration shell, facilitates the exchange of ion between the crystal and body fluids. Applied anatomy. In the genetic disease osteoporosis, which is characterized by dense heavy bones, marble bones, the osteoclasts lack ruffled borders and bone resorption is defective. So in this condition, there is defective resorption of the bone. The association of minerals with collagen fibers is responsible for the hardness and resistance of bone tissue. After a bone is decalcified, the shape is preserved, but it becomes as flexible as a tendon. Removal of the organic part of the matrix, which is mainly collagenous, also leaves the bone with its original shape, however it becomes fragile, breaking and crumbling easily when handled. Periosteum and endosteum. External and internal surfaces of bone are covered by a layer of bone-forming cells and vascularized connective tissue called periosteum and endosteum. The periosteum consists of a dense fibrous outer layer of collagen bundles and fibroblasts. Bundles of periosteal collagen fibers called perforating or sharpie fibers penetrate the bone matrix, binding the periosteum to bone. The innermost cellular layer of the periosteum contains mesenchymal stem cells called osteoprogenitor cells, with the potential to divide by mitosis and differentiate into osteoblasts. So these are osteoprogenitor cells. They transformed into osteoblasts. Osteoprogenitor cells play a prominent role in bone growth and repair. The diagram showing a section of thin portion of wall of long bone diaphysis showing both periosteum and endosteum. The externally periosteum consists of thick layer of more fibrous, dense connective tissue which merges with ligaments and the connective tissue. This is the periosteum, and internally this one is the endosteum. The periosteum, it has a good blood supply, but the cavities lined by endosteum, the marrow cavities are very rich in blood sciences and blood forming tissue. So it has a blood sciences, and this periosteum, it is having good blood supply. Endosteum. The large internal marrow cavities of bones are lined by endosteum. It is a single, very thin layer of connective tissue containing flattened osteoprogenitor cells. Just these are the small cells, and osteoblasts, which cover the small spicules of trabecula bone that project into these cavities. The endosteum is therefore considerably thinner than the periosteum. Functions of periosteum and endosteum are nutrition of the osseous tissue and provisions of continuous supply of new osteoblasts for repair or growth of the bone. Now coming towards the next topic, that is the types of bones. Gross observation of bone in cross section shows dense areas generally without cavities corresponding to the compact bone and areas with numerous interconnecting cavities corresponding to cancellous bone or spongy bone. Under the microscope, however, both compact bone and the trabecula separating the cavities of cancellous bone have the same basic histological structure. Types of bones. They are primary, immature, woven bone, mature, secondary or lamellar bone. Primary immature woven bone, they are present, for example, socket of the tooth near the surface of the suture of the skull and repair process of the fracture bone. These are the primary bones here present, socket of the tooth and near the suture of the skull. As you know about the sutures in the skull, and similarly whenever the bone is fractured, the area where there is a process of healing is occur, so their primary woven bone is present. Mature secondary lamellar bones. Cancellous bone. Cancellous bone, just like this spongy bone, this is present epiphysis of long bones. And compact bone, this compact bone, this one, this is present diaphysis of the long bone. In long bones, the bulbous ends called epiphyses are composed of spongy bone covered by a thin layer of compact bone. The cylindrical part, the diaphysis, is almost totally composed of compact bone with a thin component of spongy bone on its inner surface around the bone marrow cavity. Short bone usually have a core of spongy bone surrounded completely by compact bone. The flat bones that form the calvaria, skull cap, have two layers of compact bone called plates, staples, separated by a thicker layer of spongy bone called the diploid. These are two layers, just two layers, in between them there is a small space of spongy bone called the diploid. Primary bony tissue. Primary bone is the first bone tissue to appear in embryonic development and in fracture repair. It is characterized by random disposition of fine collagen fibers and is therefore often called woven bone. It is usually temporary and is replaced in adults by secondary bone tissue except in very few places in the body, for example near the sutures of the calvaria, to sockets and the insertion of some tendons. In addition to the irregular array of collagen fibers, other characteristics of primary bone tissue are a lower mineral content, it is more easily penetrated by X-rays, and a higher proportion of osteocytes than that in the secondary bone. So this is the primary bony tissue. Secondary bony tissue. Secondary bony tissue is a type usually found in adults. It is characterized It characteristically shows multiple layers of calcified matrix, each 3 to 7 millimeter thick, and is often referred to as lamellar bone. The lamella are quite organized, either parallel to each other or concentrically around a vascular canal. Just you can see these are the lamella around the secondary bony tissue. Each complex of concentric bony lamella surrounding a small canal containing blood vessels, nerves, and loose connective tissue is called an osteon, formerly known as a Haversian system. So, this is a osteon. The central there is a nerve vessels, blood vessels, and around this they are the concentric lamella. Just another picture of this. These are the collagen fibers orientation in these lamella. These are the lamella, circular concentric lamella. Lacuna with osteocytes are found between the lamella. These are the lacuna, and cells in between them are the osteocytes and canaliculi. These radiating from these lacuna, these are the canaliculi, which allow to cells to be in contact with the source of nutrients and oxygen in the osteonic canal. The outer boundary of each osteon is a more collagen-rich layer called the semer line. So its outer boundary is the called the semer line. These are the osteocytes and these are the canaliculi. So in the center there is a osteocytes, and space surrounding this is the lacuna. An osteon. In preparation of dried ground bone, osteons can be seen with lacuna, L situated between concentric lamella. These are the lamella, and interconnected by file canaliculi. Although it is not apparent by light microscopy, each lamella consists of multiple parallel arrays of collagen fibers. In adjacent lamella, the collagen fibers are oriented in different directions. The presence of large numbers of lamella within the differing fibers orientation provides the bone with great strength despite its light weight. Only remnants of the osteocyte in some lacuna and of the osteonic canals contents are seen in ground bone. So these are the osteocytes, these are the lamella, this is the Haversian canal H, and these are the canaliculi, and this is the lacunae. In compact bone, the diaphysis of long bone. This is the diaphysis of the long bone. Besides forming osteon, the lamella also exhibit a typical organization consisting of multiple external circumferential lamella and often some inner circumferential lamella. Inner circumferential lamella are located around the marrow cavity, and external circumferential lamella are located immediately beneath the periosteum. So this is the periosteum, outer fibrous layer, inner cellular layer, and in between this is the external circumferential lamella, and similarly here is the internal circumferential lamella around the marrow cavity, and this is the osteon, and surrounding circumferential lamella these are present. Each osteon is a long, often bifurcated cylinder generally parallel to the long axis of the diaphysis. This one. It consists of a central canal surrounded by 4 to 10 concentric lamella. This is the concentric central lamella. Each endosteum lined canal contains blood vessels, nerves, and loose connective tissue. The central canal communicates with the marrow cavity and the periosteum and with one another through transverse or oblique perforating canals, now formerly known as Waxman canal. These are the transversely communicating canals through which they are to the communicate with the periosteum and to the endosteum. The transverse canals do not have concentric lamella, instead they perforate the lamella. And all osteonic and perforating canal in bone tissue come into existence when matrix is laid down around pre-existing blood vessels. Diagram showing transverse perforating canals connecting adjacent osteons at the left side to the micrograph. Such canals perforate lamella and provide another source of microvasculature for central canals of osteon. This is central canal of osteons, and this is the adjacent transverse perforating canals. In this diagram, remodeling of compact lamellar bone showing 3 generations of osteonic eversion system and their successive contributions to the formation of interstitial lamella. Remodeling is a continuous process that involves the coordinated activity of osteoblasts and osteoclasts and is responsible for adaptation of bone to change in stress, especially during the body's growth. First generation eversion system, then there is the second generation eversion system, intermediate or interstitial lamella, and then is the third generation eversion system. Bone remodeling is continuous throughout life and involves a combination of bone synthesis and removal. In compact bone, remodeling resolves part of old osteons and produces new ones. Resorption involves the action of osteoclasts, often working in groups, or remove old bones in tunnel-like cavities having the approximate diameter of new osteons. Such tunnels are quietly invaded by many osteoprogenitor cells and sprouting loops of blood capillaries just like this, both derived from the endosteum or periosteum. Osteoblasts develop line the wall of the tunnel and begin to secrete osteoid in the cyclic manner, forming concentric lamella of bone with trapped osteocytes. In healthy adults, 5 to 10% of the bone turns over annually. So this is the newly calcified bone, is the osteoid and osteoclast tunneling into old bone. These are the mesenchymal cells present. This growing capillary grows inward. Endothelial cells are present, and these are the osteoblasts which lay new osteoid formation is occurs, and then there is a new bone formation occur. And in the lacuna with osteocytes are present. This is the old bone. So in this way, tunnels are quickly invaded by many osteoprogenitor cell and sprouting loop of blood capillaries. Thank you.